So a short little video here to highlight the difference between purine, which is A's and G's, and pyrimidines, C, T's, and U's, their biosynthesis, so anabolic sort of considerations here. So let's start first with purines. So purines, again, are our A's and our G's, okay? So I want you to sort of have a two sort of step piece to sort of think about here that's different between purines versus pyrimidines. With purines, we are going to build the base on the sugar, and we're going to have a common intermediate that we're going to see. So those are two important pieces I want you to remember, that we build our base on the sugar, right? So we're going to have a ribose sugar, and we just build the atoms around it to build either A or G, and again, that's going to proceed through a common intermediate that we're going to call inosine monophosphate. So we're going to see a common intermediate. So that common intermediate is going to be IMP, okay? So that is inosine. All right, so here's what we're going to see here. We start with ribose 5-phosphate. Remember, we're going to build the base on the sugar. So the base that we're going to build is this common intermediate called IMP. So IMP can be modified in one arm to make AMP, and then kinases can add phosphates to make ADP, and then ATP. And then the other arm over here is going to be making GMP. And further kinase activity to generate GDP and then GTP. So again, we're going to have kinase activity responsible for sort of doing this biochemistry. So we're going to have kinases here. So one of the things that we'll see, and it's very common in metabolic pathways, is we're going to have feedback inhibition. So both AMP, ADP, GMP, GDP, these can feed back and inhibit sort of this initial first step that's going to be responsible then for synthesizing these molecules. So this is feedback inhibition. Okay, so big pieces. Obviously, I want you to know who the purines are, A's and G's. I want you to think about what happens. We build the base on the sugar, and it proceeds through that common intermediate that we call inosine monophosphate. Okay, so let's talk about pyrimidines now. So really, we're talking about C and U, and as we're going to see, since we synthesize the riboflavor first, it's going to be that U that is converted into the T. So here's how this is different. We're going to build the base first, and then the very last piece is attaching the sugar. So we're going to build our base first, and then we're going to attach sugar. So the sequence of events is a little bit different, but we do have a more sequential synthesis that we have here. Okay, we don't see that common intermediate like IMP like we did in the other synthesis of purines. So here's kind of the recipe, if you will. Don't need to remember this per se, but I know you're going to be able to think about and say, hey, if we need to have a single carbon source, where can we get that? Well, bicarbonate is often a single carbon source. Amino acids are great nitrogen sources, so in this case we're going to see glutamine, and obviously we're going to need ATP to sort of help in this early step here, and again, multiple arrows here just indicating that it's multiple steps that are going to be happening here, but we're going to see in an enzyme called ATCase. This is an important enzyme because this is the rate determining step that we see for pyrimidine synthesis, and we're going to generate something called carbamoyl phosphate. Okay, and then a few more steps. We're going to be adding ribose 5-phosphate. Okay, and then that's going to generate UMP. Okay, so this is sort of where this piece becomes interesting because this is where we're going to talk about how some chemotherapy agents can sort of work. So let's continue first kind of along this pathway to make either UTP and then CTP. So again, this is sequential synthesis here where we're going to have some kinase activity generating UDP and then UTP. And it's actually one little last conversion that converts UTP into CTP, but it's kinases that are responsible for doing these phosphorylation pieces. So kinases, again, are responsible for sort of this intermediate part here. Okay, but where things get really interesting is where we talk about what happens here with UMP. So we are going to have ribonucleotide reductases, and we've talked about those already. Ribonucleotide reductases are going to make an intermediate that we don't really see used biochemically, but it's deoxy flavor of UMP. And then we're going to see methyl transfer chemistry to generate DTTP. So we're going to see here the addition of a methyl group. And if you look at the structures of U and T, and I don't need you to know the exact structures, but I do want you to know that the difference between U and T is a methyl group. So what we're going to have here is we are going to have the conversion of the ribo UMP into the deoxy flavor using the ribonucleotide reductases, and then we're going to see the addition of a methyl group and additional kinase activity that's ultimately going to generate the TTP that we need for DNA synthesis. So again, kinase activity is going to be necessary to kind of generate our triphosphate sort of species, but using sort of this piece, these two enzymes, we're going to talk about the enzyme that does this chemistry. It's called thymidylate synthase. Okay, we're going to see that on another slide here, but this is where we are going to see chemotherapy sort of agents work. So chemotherapy agents are going to kind of operate here in this conversion of DUMP to DTTP.